Okay, so we are at chapter 15, and we are finally going to talk about genes and what they are and what they do. Mendel figured out that there are instructions, genes, on chromosomes, and that's the chromosomes that are passed during sexual reproduction. So chromosomes are passed to the gametes, and then two gametes fuse to make the new individual. And so the way that a new individual is made is that the cells in the individual are expressing the genes that are contained in those cells. So we call this gene expression. That's the name of this whole process that we're going to look at in chapter 15. So if you think of genes as instructions for making something, like a cookbook, you might have a cookbook that can tell you how to make every part of a meal, but that doesn't mean you necessarily make every recipe in that cookbook. So each cell in the human body contains the same 46 chromosomes that every other cell contains. They were all copied from that original zygote. But each cell doesn't make all of the recipes that are in those instructions. Each cell doesn't follow all the instructions for all the genes that it contains. And what I'm saying is that some cells are muscle cells. So those cells only produce or express genes that are particular to muscle cells. They only follow the recipes that are important for muscle cells. Some cells are skin cells. They follow the recipes. They follow the genes or express the genes that are necessary to make them do what they need to do. So even though every cell contains all the instructions for making everything in the body, each tissue type only expresses a subset of those genes, and that's what creates the phenotype differences for all the tissues. So all the cells are genetically identical, but not phenotypically identical in the sense that each type of cell is expressing a different group of genes from the entire 46 chromosomes that it contains. So this is a really big concept, and in chapter 15 we're going to cover the basics, but you'll end with more questions than you started with, possibly. My goal would be that your questions throughout this course are now a little bit of a higher level. So you know some basics, but that brings up a new level of questioning. That's okay. I want you to know that that's okay. So, even though Mendel was kind of dancing around the concept of genes and alleles, he definitely was. Typically, we credit Archibald Garrod in 1902 with first thinking about inheritance and inheritance of alleles that cause a disease. So he recognized that there was a disease, and there is a disease called alkaptonuria. It is inherited, and it is carried on an allele that is actually recessive. So the normal allele, no disease, for alkaptonuria is a dominant allele. Most of us have that one. But if you have two recessive alleles for this particular gene, then you will have this disease called alkaptonuria. And the interesting phenotype or symptom of this disease is black urine. Black urine. And so he proposed that patients with the disease lacked a particular enzyme. And that ends up being the case. People who have alkaptonuria, their cells don't make a certain enzyme. And enzymes are the product of gene expression. So that's going to be the big concept that we're going to talk about in this chapter. In fact, all proteins, pretty much everything in the cell is the product of gene expression. And so genotype dictates phenotype because genes encode proteins. And an enzyme is a protein. Fast forward to 1941, a very famous experiment by BIOL and Tatum. They used X-rays to intentionally create mutants in bread mold. So mold is an organism. And they identified mutants that lacked certain enzymes. And so building on this idea of Garrod, they came up with the idea that every gene encodes one enzyme. Each one gene encodes one enzyme. It's called the one gene, one enzyme hypothesis. Now, it turns out that BIOL and Tatum were too narrow. They were too focused in this hypothesis. The concept really became expanded to be one gene, one protein. So each gene encodes a protein. And then we kind of expanded that idea again. But as a starting point, I think thinking of each one gene encoding one type of protein is a good starting point to think of. That protein might be an enzyme, but it could be some other kind of protein as well. So we're going to look at this from that starting point. And gene expression goes by a couple of terms. So these are all synonyms, okay? Sometimes we call it the central dogma of biology. And if you look at what those words mean, central means it's very important. Dogma means it's really important. So the central dogma of biology is like the central rule of all of biology. So that's what we're talking about here. But another way to say it is gene expression. And the central dogma of biology, aka gene expression, refers to really a couple of processes that happen inside every cell. And it's going to connect the information or the instructions that are in DNA, in the chromosome, to the proteins that are made in the cell. And so what was eventually worked out, not by BIOL and Tatum and certainly not by Garrod, but eventually worked out is that sections of DNA on the chromosomes are copied. And in this chapter, we're not talking about copying the chromosomes in preparation for a cell division. We're talking about copying from a DNA template to make a copy, but make that a copy fully in RNA nucleotides. So here, if you copy this DNA template here, you have a G and then an A and then a T and then a C and so on. If you copy it, when you look at this complementary strand that could be built complementary to the DNA, you would have an RNA that says C, U, A, G, and so on. So these are complementary base pairs. Remember that in RNA, we have uracil instead of thymine. So anywhere you would want to put a thymine, T, instead put a U. So this would be the RNA copy of this gene. So this would be the gene. It's a section of DNA. And this would be the RNA that's copied from that gene. And the process of copying from a gene to make an RNA is called transcription. So transcription is the first step of gene expression, copying from DNA to make a full RNA molecule. Now, one thing about transcription is we're not copying the whole chromosome. We're just copying a section within a chromosome. So a gene is just a section, a short section within a chromosome, not the whole chromosome. And so making these little short RNA segments. But the gene has information, and so when we copy it into the RNA contains a version of that information. And that information is used to make a protein by a second process called translation. Translation looks at the information in the RNA and builds a particular kind of protein. So each RNA would have a different sequence of nucleotides based on the sequence that was found in the gene, and that tells the ribosome how to build the protein. Remember that ribosomes are the organelles that build proteins. They connect amino acids together by dehydration synthesis, and that builds proteins. Once a protein is built, then it folds into its secondary and tertiary and quaternary structures. Remember, shapes. And then that protein, because of its shape, does a certain job inside the cell. So, central dogma of biology includes transcription followed by translation, gene expression, also involves transcription. So these mean the same thing. Transcription first, the copying from a DNA template to make an RNA. So the product of transcription itself is RNA. But then translation takes an RNA and a ribosome and uses that combination to build a protein. And so the product of translation is a protein. So you have these two processes back to back. And that's called gene expression. And what happens is if something is wrong in the gene, if there's a mutation in the gene, like this is supposed to be a C, but it somehow got changed to a G, then that means that in the RNA, there would be something that's wrong. And then in the protein, there would be something wrong. So if you have a mutation in your gene, then that might mean that your protein that's being built based on that information is wrong. It might not form the correct shape and might not be able to do its job. And that's really what happens in alkaptonuria. There's a mutation in the gene. It produces a copy in RNA, but the copy, when it's read by the ribosome, has the instructions are messed up, so the protein that's built doesn't do its job. And in the body, you end up with this phenotype or this symptom called alkaptonuria, black urine. All right. So that's an overview of what this whole chapter is about: gene expression, the central dogma of biology, and the processes are transcription, which produces RNA, translation, which produces proteins. And I apologize that the names of these processes both start the same, transcription and translation. I didn't pick the name. So we want to think of transcription as how cells make RNA. This is how cells make RNA. And translation is how cells make protein. And in gene expression, you have transcription first and then translation. So it's a sequence, one and then the next. So transcription makes RNA, and then the RNA has the information. It finds a ribosome, and the ribosome reads, we think of it as reading the RNA to make a particular kind of protein or polypeptide. So I pointed out that RNA contains uracil as one of the nitrogenous bases, not thymine. Oops. And there are actually lots of kinds of RNAs. We're going to be focusing on one called mRNA, but any RNA that you ever see mentioned is produced by transcription. So all RNAs are made by transcription. Only mRNAs participate in translation, however. Well, there's a couple of supportive RNAs too, but for the most part, mRNA is the main star of translation. So all types of RNAs are made by transcription, and some of them do things on their own, separate from translation. But there's a few types of RNA, especially mRNA, that are involved with this second process called translation. So that gives us a good starting point. I'll show you on the next slide some different RNAs that exist. I don't know if you know any kinds of RNAs, but the three that are involved, they're made by transcription. but these are involved with translation. So only these three are involved with translation, whereas all of these are made by transcription. So all of them are made by transcription, but only these first three have go on to the second process that we're talking about in this chapter called translation. All right. So we will take a break, and then we will start talking about transcription first.